Hello, my name is Stéphane Vidry. I'm the Executive Director of ILSI Global, and I'm happy to welcome you for this session 2 on the microbiome beyond the gut. It is my pleasure to welcome the chair of the session, Dr. Christian Hoffmann at the University of Sao Paulo. Dr. Hoffmann research focuses on the interplay between the gut microbiome, diet, and immune modulation, how it changes as we shift our lifestyle and how to make use of this relationship to promote human health. And we thought that he would be the perfect experts to indeed share this session about the microbiome beyond the gut. So Chris, the floor is yours.
Thanks so much, Stefan, for this kind introduction. It's a pleasure to chair the session. When I was invited, I actually asked what brought this all together, and it was the idea to actually show a case microbiome work beyond the gut and potential interactions with the human gut, which is one of the central focus that ILSI has addressed in the previous years. But there's also this need to address other areas of microbial ecology that actually can affect human health. So we'll have a very nice overview from this diversity. Today we'll see a bit of soil, a bit of plant, and then we'll finish with 2 systems from the human body. So it is my pleasure to address our first speaker, Dr. Linda Kinkel. She's a professor of plant biology at the University of Minnesota. She researches ecology and evolutionary plant biology of plant-associated microbes. Her interests are in managing these beneficial microbes in the soil to enhance plant health and perhaps increase productivity. Today, she'll share with us a talk entitled Soil Microbes and Crop Health. Are soil microbiomes the key to sustainable cropping? Dr. Kinkel, it's a pleasure. And just reinforcing to our audience, please use the questions panel and we'll address the questions at the end of each one of the presentations. So please, Linda, you have the floor.
Thank you. And thank you to the organizers. I'm pleased to be here today and talk a little bit about microbes and crop health with you today. I think we're all aware of the profound challenges we face in feeding a world that will have a population well above 9 billion by 2050. And even at current population levels, we have a significant challenge to reduce global malnutrition. Estimates are that we'll need to increase food production 20 to 70% to meet the need by 2050. So this raises a question. Beyond the challenge of food waste, which we're becoming much more aware of, there's multiple significant factors that limit crop yields at the field level. And these include inadequate moisture, lack of nutrients. Oops, I'm seeing everybody's faces now instead of my screen there. Inadequate moisture, lack of nutrients, suboptimal soil conditions. Soil degradation is a significant challenge globally. Pests and pathogens reduce yields, and climate change is beginning to represent a significant challenge. Suboptimal temperatures, either too high or too low, have significant negative impacts on crop yield. Traditional responses or solutions to these are inputs. That is, irrigate, add inorganic fertilizers, add pesticides. This is our traditional approach. And in fact, we've bought ourselves many decades of increased productivity through inorganic inputs. Yet these inputs all have consequences, mostly negative for our environment. We recognize that the challenges of water shortages globally and increasing soil salinization due to over-irrigation, impending global phosphorus shortages are real. Inorganic soil amendments tend to be quite costly and not available in developing countries, and they tend to also have limited effectiveness. And finally, we know that the classes of pesticides on the market have negative impacts for non-target organisms, including humans, with pesticides in our pesticide residues in our food. So these negative consequences have led many of us to begin to say it's time for a fundamentally different approach. That is, we had the green revolution of the 60s and 70s. Is the time now coming for a microbial revolution in agriculture? That is it time for us to begin to explore how we can optimize plant and soil microbiomes to support global food production? And to what extent is this a practical strategy for us to close the yield gap, to reduce reliance on external inputs, to restore degraded lands, and to expand productivity and capacity of marginal lands globally? Now, we all know over the past years, it's clear microbes are everywhere. And this year's pandemic is especially a great reminder that microbes are everywhere and they will find you. Plants evolved and exist in intimate symbiosis with microbes. Microbes predate plants evolutionarily. They've always been together. And plants and the ecosystem services they provide globally rely profoundly on the services that the microbiomes provide to them. Crop yields are influenced by microbial partners, but the reality is our history of agriculture is focused predominantly on pathogens, on one very tiny slice of the microbial world. But briefly, to let you know, the densities and diversities of microbes that are in close association with plants is enormous. So what is the potential here? In soil, in the vicinity of a crop plant root, there can be up to 10 to the 10th cells of bacteria in a single gram of soil, the tip of your pinky finger, 10 to the 10th cells. Inside plants, bacteria can proliferate dramatically. On the leaf surface, we see bacteria in association with the stomate. Here's a leaf surface, we see fungal hyphae, yeast cells, and bacteria. Plants are bathed in microbes in the soil and on the plant surface. Looking at the fungi, which can be present at densities of 10 to the 5th to 10 to the 7th cells per gram, and this beautiful close-up of soil, look at the fungal hyphae ramifying through this soil. The fungi are critical to maintaining soil structure, soil aggregation, and you can see here in this red pine seedling, whose root system is encompassed by these thick roots, but is colonized by a fungus that expands the effective root system up to 80%. So fungi, and finally viruses. Viruses, of course, are not free living, but they're densely colonizing, if you will, soil, both due to their presence in plant tissue, but also inside fungi and bacteria. There can be 10 to the 9th virions per gram of soil. So plants are living in an incredibly rich array of microbes in that soil community. So what can these microbes do beyond the pathogens, which we've spent most of our intellectual capital on for the past century? Beneficial microbes that are common in soil can help plants acquire nutrients. In addition to expanding root capacity, microbes can fix nitrogen and make it available to plants. They can solubilize phosphorus, also making it available to plants. Microbes can protect plants against pests and pathogens by directly antagonizing and killing pests and pathogens. Microbes can also turn on plant resistance to help plants keep from becoming infected by other microbes. Microbes can help plants tolerate environmental stresses, including salinity stress, drought stress, temperature stress, and environmental pollutants. And finally, microbes can help plants grow better. Many soil microbes produce plant hormones. When those microbes are present, plants grow bigger, stronger, and faster. So we recognize the significant potential that microbes have to help plant productivity. So this isn't a new phenomenon, right? How do we capture the benefits of microbes to enhance sustainable production? Well, over the past decades, we've tried many different things. Inoculants, inoculate microbes. Here's a microbe that can do a good thing. Let's inoculate it. We've been doing that with Rhizobium for decades. More recently, there are many, many different microbial inoculants on the market. Alternatively, we can manage indigenous populations. That is, every soil has microbes with tremendous potential to support plants. How do we enrich and enhance the capacity of the already existing communities to provide benefits? And finally, there's active efforts among plant breeders to manipulate plant genotypes to support colonization of beneficial microbes. Today, I want to go through an example talking about a strategy for developing a mindset for managing indigenous populations to support beneficial functions. Now, this is important. I have to note that we've been managing soil microbiomes as long as we've done agriculture. Every time we plow or irrigate or add NPK, we're changing soil microbiomes, but we have virtually no insight into what those impacts are. But what we need to do as we move towards a soil microbiome management, we have to fundamentally change the way we think about these management strategies from controlling a population, that is, let's minimize a pathogen, to understand the entire network of organisms that are interacting in that soil. And there's fundamental questions we don't know anything about. That is, to date, there still is limited information on what the characteristics of a good microbiome look like. What should a microbiome look like to optimize productivity? And how can management achieve that outcome? In our research, we have taken a full head-on strategy to try to address this question, looking at a naturally occurring disease suppressive soil. This is a soil that after 34 years of potato monoculture, so for 34 years, potatoes and only potatoes were grown in a small plot of land. And this was because it was a plot that had very, very high levels of two important potato diseases, potato scab and verticillium wilt. So this plot was useful for breeders to screen breeding lines. If they planted potatoes in that field and they came up with disease, they threw them out of the breeding program. But after 30-odd years of continuous potato monoculture, disease disappeared from that field. Even the most highly susceptible potatoes would come out of that field with no disease. When I started my career at the University of Minnesota, the first thing that I did was to say, what happened? So I went to that field to try to understand why that soil suppressed pathogens. And the first thing we found in that soil was lots and lots of bacteria in the genus Streptomyces. And in the laboratory, we found that those Streptomyces were outstanding at inhibiting lots of different plant pathogens, including bacterial pathogens, oomycete pathogens, fungi, and even nematodes. And so we did the simple thing, which was first, let's inoculate. We took some of the best killers, the best antibiotic producing isolates. We inoculated them into a potato field in April at planting. At harvest in September, we had inoculated potatoes that came out quite lovely, and the non-inoculated with the characteristic symptoms of the diseased potato scab. We've inoculated in many, many different crops. Here's an example of an inoculation on alfalfa. Here we had soil that's naturally infested with a pathogen, Phytophthora. These are non-inoculated pots. And these are pots that at the time of planting of the alfalfa seeds, each pot received 1 milliliter of 10 to the 8th spores of one of our really good antagonistic Streptomyces. And 28 days later, you see the benefits of that inoculant. But realizing that I could spend an entire career finding isolates from that soil and trying to inoculate them in other locations, the more important question, and the question that's the frame shift, let's move away from an inoculative vision to understanding what happened in that community. That is, why is that soil suppressive? Or more to the point, how did it get that way? How did that soil microbiome move from highly disease-rich, intense disease to no disease? Now, unfortunately, that field was initiated back in the 1940s, and we don't have soils that go back that far. But on the research station adjacent to the suppressive soil, we have a field that remains fully conducive to both potato scab and verticillium wilt. So these two soils are about 10 meters apart. These two fields, they differ in cropping history. They're indistinguishable in NPK, but one is suppressive and one is conducive. So we compared the Streptomyces communities. The question was, is this something comprehensive about the Streptomyces communities that is in fact associated with disease suppression in a comprehensive way? And we asked 3 simple questions. We said, maybe suppressive soils are suppressive because they have higher densities of Streptomyces, and that was true. We had significantly higher Streptomyces densities in the suppressive than in the conducive soil. So more Streptomyces, maybe they're better at killing pathogens. We randomly collected 300 Streptomyces from the suppressive and 300 from the conducive soil, and we looked at the ability of every one of those Streptomyces to kill 21 different plant pathogens. And we found for every pathogen, a greater proportion of Streptomyces could inhibit that pathogen when the Streptomyces were from the suppressive soil. And the mean inhibition zones, the zone sizes of killing, were significantly greater against every plant pathogen for populations from the suppressive and the conducive soil. So there's more streptomyces, they're better antibiotic producers. And in fact, they produce a higher diversity of antibiotic inhibitory phenotypes. So you might say that's interesting, but does it tell you how? And we think actually these data suggest an important way of thinking about what happened in that soil. So potatoes were grown for 34 years. Where do potatoes put their carbon? They pump their carbon below ground in big juicy tubers. And they have root systems that actually turn over fairly rapidly. This is a great way to feed your saprophytic community, and streptomyces are saprophytes. So we hypothesize that long-term potato monoculture enriched the streptomyces densities. There are more of them. Now you have lots of streptomyces competing in the soil at much higher densities under these high density, highly competitive settings. Antibiotic production should confer a greater fitness benefit than in a lower density setting. And this should generate a density dependent selection for antagonistic or antibiotic producing streptomyces, which should yield this increased frequencies and intensities of inhibitory phenotypes. So now you have a rich streptomyces community with lots of antagonistic activity. Yet think, if all those populations are producing the same antibiotics, they're wasting their time. Yet that clever streptomyces that is able to produce a novel or a rare antibiotic will obtain a greater fitness benefit from that rare antibiotic than from a common antibiotic. And this is a model for frequency-dependent selection, that is to diversify within that community the distinct antibiotic inhibitory phenotypes. And so we hypothesized that this long-term monoculture induced straightforward density and frequency-dependent selection that generated a disease-suppressive soil. So we can rephrase, we can reconceptualize this model as a basic story about coevolutionary selection. We think of soil communities with an evolutionary potential that's determined by the density and the diversity and composition. But we highlight in our predictive model microbial species interactions. We argue strongly that microbial competition is a critical driver for this functional outcome we care about, this disease suppression. And in particular, we predict that competition can generate an antagonistic arms race. There's a plate of cookies, I kill you, that imposes tremendous, I get all the cookies, but this imposes tremendous selection pressure on these coexisting populations to become resistant and generate their own weapons. This arms race is what we hypothesize generates that highly diverse, highly antagonistic community. Yet in a lot of data that I'm not going to have time to show today, we also show that communities might undergo a distinct coevolutionary trajectory. That is, while antibiotic competitive interactions are extremely valuable for disease suppression and pathogen control, competing microbes might have a different coevolutionary outcome. That is, there's a plate of cookies. Certainly, if I produce antibiotics, I get all the cookies. Yet antibiotics are metabolically costly. And it may be that if there's chocolate cookies and peanut butter cookies, my fitness may be optimized if I eat the chocolate cookies, you eat the peanut butter cookies, somebody else eats the fig Newtons, and collectively this niche differentiation in a soil microbiome can also lead to stable coexistence. But the community in that case has a very different functional outcome. That is, rather than antagonistic, you have populations that are highly growth efficient on a small range of substrates, yielding a community that's very efficient and effective in nutrient cycling. So these functional outcomes reflect really different species interaction trajectories, which raises the next critical question: what determines which trajectory a soil microbiome might follow? And I'm skipping a little background information. In fact, naturally occurring disease suppressive soils have been documented for nearly a dozen agricultural systems globally, and are virtually always associated with long-term monoculture. We think that monoculture, if you think about monoculture growing the same crop year after year, it's just like you only have one kind of cookie. We argue that monoculture might be a really good way to force an arms race coevolutionary trajectory. While in contrast, polyculture, this idea of lots of different resource availability, establishes a habitat in which niche differentiation is possible. And so we have three testable hypotheses embedded in this model. One is that monocultures will generate an arms race and antagonistic coevolution. and pathogen suppressive communities. Polyculture will generate niche differentiated microbiomes, and that it's a difference in the nutrient diversity or nutrient richness between these that determine that outcome. We actually had, we're studying some long-term field plots at a National Science Foundation long-term ecological research site, which allowed us to test these three hypotheses. So the first question was, are Streptomyces populations in long-term plant monocultures more inhibitory than populations in polycultures? We looked at 4 different plant species, 2 C4 grasses and 2 legumes that have been growing for 14 years in a monoculture or in a community of plants 4, 8, or 16 species rich. We've looked at those microbiomes from many different perspectives, and today I'm just going to give you a tiny snapshot, focusing on, first of all, are populations more pathogen suppressive in monoculture? And what we do is we do a soil dilution. We wash the soil, we plate it, we let it grow for 4 days, and we overlay with a pathogen. So we tested against 5 different important fungal and bacterial plant pathogens, and we count for every community what are the number of inhibitory isolates and what's the mean inhibition zone size. And we compared this across diversity levels, that is with 4 different plant species growing in monoculture versus species up, communities up to 16 species. The mean proportion of Streptomyces that are inhibitory against pathogens is significantly higher at low than at high diversity. And the mean inhibition zone is also significantly greater in monoculture than in high diversity communities. That is, monocultures do support significantly more inhibitory populations. The second prediction was, of course, that populations in polyculture should be more niche differentiated. And we determined nutrient use for every isolate on 95 different carbon substrates. And then we measured, and I apologize, we actually measured niche overlap, which is, of course, the inverse of niche differentiation. Niche overlap is less in polyculture than in monoculture, which tells us that the polyculture populations are more niche differentiated. They tend to use different nutrients. So the core finding that we think is critical in this work was that microbial species interactions are clearly driving the functionally different outcomes in these monoculture and these polyculture communities. Now, this doesn't mean the plants are not important. Remember, the third prediction was that plant diversity would influence soil carbon richness. Really, that these interactions are mediated by the richness, the number of different kinds of carbon. Is there one kind of cookie or are there many? And so the final thing we did is we looked at carbon compound richness using pyrolysis GC mass spec for one of the grass species growing in monoculture and in polyculture, and one of the legumes growing in monoculture and polyculture. And the simple outcome is that soil carbon richness is significantly smaller in monocultures than in polycultures. So collectively, these data show that the microbiomes and their functional capacities are exquisitely adapted to that local environment, but critically, it's interactions across multiple scales, that is, microbes interacting with one another in the rhizosphere of an individual plant host embedded within a community with its own characteristics, monoculture or polyculture. All of these factors together are determining that functional capacity of the soil microbiome. And so thinking of microbiomes, I think one of the critical things that we need to think of with the managing soil microbiomes is moving away from our strategy to manage a population to actually consider managing the biotic interaction. That is, managing populations has been our historical legacy. Let's kill the pathogen, let's inoculate one rhizobium. But instead, by untangling these communities, can we actually manage the ecological and selection dynamics that determine functionality? And in doing this, I have no illusions this is simple. We have to balance a diversity of functions. Do you want highly nutrient cycling efficient? Do you want antagonistic? Do you want P-solubilizing? Understanding these different interactions that generate these functions and recognizing you might need something very different from a high input dependent potato system versus a grassland grazing system. But microbiomes offer us the potential to optimize for sites. This is not a one size fits all approach. And getting close to the finish, I want to note that this session is a lot about the microbiome. But we have to think bigger than just the microbiome. The phytobiome collectively, the plant and the microbe in the soil with the animals and the climate and the nutrients. It's an integrated unit of production. And microbiomes in particular are not only interacting with the plant, they're actually critical to mediating nutrient availability, soil structure, and even climate through ice nucleation activity. And so beginning to integrate microbiome thinking, bigger thinking into this broader unit of production is critical for agriculture moving forward. I was asked to touch on what we need to build the science, and I just want to make a couple of key points. We need much more big thinking, collaborative science and public-private partnerships. And in fact, there's a tremendous amount of activity in this area. I'm part of the International Agricultural Microbiomes Research Coordination Network that's seeking to integrate research projects globally. The International Phytobiomes Alliance is also playing a leadership role in this area. The EU Microbiome Support Project and many, many international projects, especially focusing on soil microbial databases or soil microbiomes, including the African Soil Microbiome Project. So there's a lot of activity here. We need to do a little bit of better job than we're doing bringing it all together, but the efforts are moving forward. Microbiome-focused databases are a critical need globally. The US investment through the Department of Energy in a National Microbiome Data Collaborative, I think, is a really bright spot, and I'm excited to see where that goes. And the National Academy of Sciences of the US is just going to be putting out a white paper on the need for a publicly accessible soil database that has the rich content that we need to manage microbiomes. And finally, data and metadata standards, as in all fields, are critical for progress. And moving forward, it's a great time to be a microbiome scientist. The next decade is going to be rich with discovery. We still don't know who and where and when and what. What are these microbes doing with what other microbes, where and when? And what are the higher order structures or dynamics that we need to be able to capture for benefits? We also have to be committed to invention. We need better conceptual frameworks and predictive models for thinking about microbiomes in soil. We need more creative and complex analytics, and AI and machine learning are becoming incredibly critical to this. And we need inventive ways for translating to management. I should note that translation is advancing rapidly, probably a little bit faster than the science itself. There's huge investments by major agricultural companies, the big players. There are over 200 agricultural startups presently active with microbial inoculants. And in fact, efforts in the developing world, I think, are going to be especially significant to moving the needle over the next decade. And collectively, I do see a future in which microbes and soil microbiome management will play a central and really significant role in securing an adequate, safe, and sustainable global food supply. Finally, I want to thank the wonderful collection of colleagues I've had the opportunity to work with over the decades, and funding, especially from the the USDA and the National Science Foundation for supporting our work. Thank you.
Thanks, Linda. Great talk. We have here a couple of questions coming from the audience. So excellent. The first one is, which actually I thought about it as well. What happens when the farmers burn the residue that's actually left over in the field? So are there changes in the microbiome, or are those affecting soil health? What are the consequences for the soil health? Can you comment on that?
It's actually a really great question, because if you think about, as we've moved away from intensive tillage to no-till, keeping residue has become more attractive, but it can also be a real problem for getting soil temperatures warm enough for planting the next spring if you have a heavy residue layer. And so burning residue as a removal strategy can sometimes be attractive. It doesn't have a really dramatic impact on the soil microbiome. Those burning events tend to be quick, and they don't really change soil temperatures. The question is, so one thing is the burning itself problematic. The other is the lack of incorporation of residue a significant negative factor, right? Because you're not feeding the microbiome if you don't incorporate the residue. We just are finishing up a really interesting study of some 62-year monocultures where we had residue incorporation, NPK, both of those are plus and minus in a factorial. In fact, residue surprisingly had little impact on moving the needle in soil carbon. NPK had a dramatic impact, which surprised us a little bit, but I think it's the labile exudates from the roots that actually are really important in driving that soil microbiome and its capacity to sequester carbon and maintain high carbon levels in soil. So I can't give you a simple answer, but I don't think burning is going to have a more significant effect than just removal of residue.
Okay. Our next question here is, some years ago in an ILSI workshop, it became very clear that we're not there yet as defining what is a healthy microbiome. We just don't have enough data. Is this the same thing for soil health, or are we closer to actually saying this healthy soil microbiome, this is unhealthy soil microbiome? Can you comment on that?
I'm thrilled to have that question, because I have to tell you, when we go to ag meetings and we hear about people talking about the human microbiome, we always go, that's so simple. What do they have, 1,000 species? We've got tens of thousands. So we always pat ourselves on the back for the complexity of our systems. Do we know what a healthy soil microbiome is? No. But I think that some of the big projects are underway. I'm part of a project that goes from the West Coast to the East Coast, the entire northern tier states. We have 10 states of potato production where we're doing coordinated experimentation and deep dives into soil microbiomes over time and space in potato production. I think it's going to take that big scale thinking and really rich data sets to begin to think about what a healthy soil microbiome looks like, and in particular, a soil microbiome that has supported a healthy and productive crop. But I don't think it's going to be one answer. I think it's going to vary with crop and soil type and cropping history, and that's where the complexity and AI and machine learning approaches, I think, are going to be critical for us to pull the pattern out from all that noise.
Right. We have space for a couple more questions. I'll select them here from our questions being asked. So there's a question here related to biofortification. So is there a lot of interest in biofortification of plants to increase their micronutrient content? How can soil microbiome affect the nutrient content of plants for human consumption? Could you comment on that for us?
I don't have really good data on that. We have been looking in a prairie system about at the effects of NPK on nutrient status of the leaves and the seeds, but I don't know that the relevance of that to our food crops and their nutritive value. I don't know what limits nutritional value or accumulation of high vitamin content or other valuable attributes of fruits and vegetables. So I just don't know the answer.
I'll finish here with a nice question that perhaps links a little bit with the future presentations. So most fruits and plenty of vegetables actually are consumed raw. So what does this mean for the intake of these plant microbes if we start fortifying them, for example? What do you think? What are your thoughts on that?
I think that there's very little downside. Of course, there are the foodborne pathogens, but most or many of you are probably aware of the stories that production of beer in Egypt that actually was filled with streptomyces could have been a way of self-medicating. I love the idea that when I eat raw potatoes or carrots that I'm consuming streptomyces. So I think that we've been eating raw vegetables for generations. I worry more about pesticides than the microbes in general, but with the big caveat that salmonella and some of the foodborne leafy green pathogens are pretty nasty.
Of course. Well, this is about the time that we have. We should be moving on. So thank you so much again, Linda, for your talk. Great talk. We'll go for our second speaker. Professor Brajesh Singh hails from the Hawkesbury Institute for Environment and the Western Sydney University. He, of course, has a very, very large time difference. So he recorded his presentation earlier for us, and we'll actually present it now for everybody. As such, we will not take questions for his presentation, and then we'll move on with our session. So, Stefan, I guess you could start now. I'll just say a couple, prepare yourself to start. I'll just say a little bit about Professor Brajesh's work, He's a soil ecologist, and his research explores the relationship between microbial diversity and ecosystems functions, and how natural and anthropogenic pressures actually affect these relationships. One of his goals would be to eventually achieve agricultural productivity and environmental sustainability at the same time. We know that today this seems to be a little bit against each other. With that said, his talk today is entitled Microbiomes, Ecosystem Functions and Sustainable Agriculture. And let's see what he has to say.
Good morning or good afternoon, everybody. Let me start with today to apologize for not being presenting live. Personal circumstances prevented me to come along. I would like to thank the organizers to ask me to come and share some of the work I'm doing in this area. As you can see, the presentations I'm going to make today is on the microbiome, ecosystem functions, and sustainable agriculture. Let's remind ourselves the soil and the soil microbiomes, what are the key ecosystem functions and services they provide. For the human point of view, one of the key ecosystem functions is crop productions. And that is being influenced by a number of processes, including the availability of plant nutrients, plant available nutrients, which is a link to enhance biological activity in the soils and increase cation exchange capacity. The other key functions is filtrations and the buffering. Soils play one of the most important role is filtering the water and buffering the water in the soils that is available for the plant uptake, and as a result the primary productivity, but also buffering and filtering for the better groundwater, which then ultimately supply back to in the stream and provide the water for the agriculture, the irrigations, but also the drinking and other recreational purposes. Soils have a critical role in the climate regulations, mainly through the emissions and mitigations or the absorptions, consumptions of the greenhouse gas And all these functions are mainly carried out by the soil microbes. But there are other drivers of the research in the microbiome, soil microbiome and the plant microbiome, including the requirement to increase the food productivity productions by 50 to 70% by 2050. Another driver is that the farm productivity has been in a structural decline since last few years, particularly in the developed country. What that means, the additions of additional inputs, the fertilizers or the pesticides, does not result into proportions increase in the productivity. Agriculture is now being increasingly called for to contribute towards climate change mitigations. There have been a loss of agrochemicals and the plant breeding program in terms of productivity gain. And there is negative impacts of the quality of the human health. But there is, I guess, there is a two type of the rationale. One is a policy top down, that where the government and intergovernmental organizations requires the sustainable productions, but also the bottom ups, where the consumers are demanding for the quality food, but also the environmental protections. So low use of agrochemicals, particularly the pesticides in the agriculture production system. If you look at all the analysis that has been done over the last couple of decades, that where this gain of productivity at the same time minimizes the environmental protections, challenge can be made, and comes from two aspects. And this is a diagrams mainly produced by Professor Fuso Chang of Chinese Agricultural University in Beijing, where they suggest that the increasing soil health alone can increase the farm productivity by 15 to 20%. And if we combine that the soil health with the resource efficient crop varieties, then we can gain 30 to 50% without further increase in the resource input in the soils. So it's contributing, increasing the productivity without further damaging our environment. And I will argue that both of these requirements, soil health and resource efficient variety of crops, will require the explicit considerations of soil and the plant microbiome. Another, the driver, is that the microbial product is the market itself is a driver. If you look at the global perspective, the microbiome contributions to the human economy, it sits around $1 trillion. And this does not include a lot of antibiotics that are mainly produced from the microbial communities. Then, if you just look for the agriculture products, it sits here. It is a biological product. It's not just a microbial product, but it's total biological product. It sits around today around $5 to $6 billion. Out of that, the 4 billions are the biopesticides. Out of half of that, more than half, 60% of that agriculture products are the microbial in nature. The microbial product in agriculture is expected to increase to 12 billion within the next 5 years. And so the increase in their value is 3.5 times faster than the chemical pesticides. Agrochemicals today sits very high in terms of the dollars. Its annual sales is around 250 billion. Out of that is 55 billion is pesticides. And it is estimated that in the very near future, the microbial pesticides will be similar market size as the chemical pesticides. So the market itself is driving the research and innovation in this area. So in my presentations, what I'm going to cover very briefly of 3 aspects. The soil microbial diversity, particularly focusing with what we call the most wanted list or the most dominant species globally, microbes, and what role they play in the ecosystem function. So providing some evidence in this area. The second point, then I will talk about the plant microbiome and sustainable agriculture. And the third, I will end my presentations with a few of the slides that talk about a global initiative that we launched to address some of the gaps in the fundamental knowledge in the plant and soil microbiome that we need to be addressed in order to advance this discipline and advance this industry. So let us start with the first, which is the dominant microbial communities. If you look at the ecological, you will see that I will use the words ecological principle a lot in my presentations, all through these presentations, because I believe that if there's ecological principle exists, that has to be unifying in most cases, not in all cases, but in most cases. So if there is a ecological theory, it should be applicable across the different organisms. And from the plant ecology and the animal ecology, we have a good understandings that there's hundreds and thousands and hundreds and millions of species of plant and animals. But if you look at the global distributions, only few species really dominate globally. And we thought if this is the case in the plant and animal, you expect similar sort of dominance in the microbial communities. This is one study that is carried out by the Manuel Delgado Macarizo. He was in the postdoc my lab, now is a well-known scientist himself in Spain, where we sampled across the globe. You can see that where the sampling has been done in the slides. What we did is to look at the microbial communities, both the bacterial and the fungal communities, and measure all other aspects of the ecosystem property. That's where it comes from, is temperate forest or the tropical forest, boreal or subtropical, agriculture or dryland, and look at the multiple functions as well in those soils where those samples were collected. So the first things to look at is is there a dominant species of, here in case it is a bacteria. And so we define the dominant species, again taking some knowledge from the plant ecology that is present in the most of the samples, so 75% and above number of samples, and it is among the top 50 in terms of the relative abundance. What you see in this diagram below, there is only 2% of OTUs, or I guess a lot of people will sell at the general and species level resolutions. Only 2% of the total, which is equivalent to 551 phylotypes, was the dominant microbial taxa across the globe. But on the right side, if you look at the same pie chart, you'll see that those 2% account for the 41% of the relative abundance of the total bacterial community, giving the evidence of the presence of the dominant bacterial phylotype across the globe. We went on to see and looked for the fungal dominant species. Again, you see there is only 0.1% that was dominant, but they accounted for the 18% of relative abundance. And what that then allows this sort of analysis is to predict the distributions of this microbial community across the globe. And here on the right side, you see that for the fungal sort of things, fungal communities, we predicted that this is where fungal species are distributed across the different ecosystems, the forest ecosystem across the globe, drylands, and the magic forest ecosystems. The other utility of these approaches is to predict the economically important microbes. In this case, we look at the distributions of the soil-borne fungal pathogens response to global warming. And so what we do, we look at the distributions of microbiomes across the globe, the soil-borne fungal pathogens, and we look at which are the key drivers. And what we found is two important factors: the moisture level and the soil temperature. And then we use the IPCC model to look at what will happen in 2035 and 2050, and projected what will be the distributions of those fungal pathogens. You see, this is Phoma, Venturi, and Fusariums, well-known taxa in terms of the fungal pathogens. And you see that what you see that instantly, the most of the species, obviously the data here is only for 3, but we've done a lot more analysis for like Alternaria, Rhizoctonia, and so on and so forth. But what you see on the middle side of it, projected in 2050, you'll see that the abundance of these organisms or plant pathogens will dramatically increase under the projected climate change in 2050. But these are the projections, these are the modeling. People always ask that we need some experiment evidence. So what we did, that we took the samples from the long-term climate change experiments, and where we look at the control, which is ambient temperatures and the warming temperatures, and look for, using this times the qPCR, to look for the abundance of these organisms, and relative abundance of these organisms. And what you see, that is consistent with our projection. In almost in every case, the relative abundance of these fungal pathogen phylotypes was increased by at least a fold under the climate change within the 10 years of the warming experiment. So that's the utility. So you can, this is just a soil-borne fungal pathogens for the plant. You can do the same things for the bacterial pathogens, for the human pathogens and animal pathogens, and so on. Second aspect is that one of the real questions have been in this, the microbial ecology, is that because microbes are so diverse, if you lose some diversity, will really have impact on the ecosystem functions. And the second question has always been the upscaling, because microbes are microscopic, so can that have impact on the ecosystems in the global level? And this is the study we done a lot earlier in the dryland. And here you see that there's two sort of database. One is a dryland, global dryland, which come from the Biocom experiments. And there is another database, which is Scotland, where I was before I came to UK. And we'll look again the bacterial and fungal community, but also look for the 16 functions we measured. And then we take the indices of that 16 measures into one measure, what we call the ecosystem multifunctionality. And so so they did done by different methods. One approach is averaging those functions into one indices. And this is a structure equation modeling that is done to link the microbial community. And we chose these two data sets again to define that if there is a relationship between microbial diversity ecosystem functions like other organisms, we know there is for the plant diversity ecosystem functions, then it has to be consistent in the different ecosystems types. And these drylands and the Scotlands are really contrasting ecosystems. In fact, I would say the orange and apple drylands are really limited with soil carbon, high pH. It is a very low carbon, while most of the Scotland soils have a very high rainfall, very high organic matter compositions, and very acidic soils. But you see in both these cases, ecosystem multifunctionality was directly linked by the microbial diversity. It is almost 0.4% variation in the ecosystem multifunctionality was explained by the microbial diversity in the dryland, and similarly the ecosystem functions in the Scotland, saying that the impact of the relationship between microbial diversity and ecosystem multifunctionality is consistent irrespective of the ecosystem types. And after that, we have shown that in multiple ecosystems, including Australia, agricultural ecosystems, and so on and so forth, but also at all levels, from microscopic level to the global level. So that was the relationship between ecosystem function and multifunctionality. The second part of my discussions today, or my talk today, is the agriculture microbiome. As I said, there is a lot of demand to explicit considerations of the microbiome and development and harnessing of microbiome tools to increase the farm productivity to meet multiple sustainable development goals of the United Nations. And this is because we have very good understanding now that the soil and plant microbes play a very critical role, particularly the soil microbes, in terms of the provision of the nutrients through the fixations, nitrogen fixations, and nitrogen and phosphorus mineralizations and productions of the phytohormones. But also, both the microbes of the soils and the plant microbes play a crucial role in the plant defense against the pest and the pathogens. The other aspects is that they're having, I guess, so there is a lot of push, as I discussed briefly, both from the policy point of view, but also from the consumer demands. And this has resulted into some of the real, driven by some of the real translational success stories. I guess you guys probably heard that the European Union's Green Deal requires that 50% of the chemical pesticides and 30% of chemical fertilizers to be reduced in the use in agriculture by 2030, 9 years from today. And that's probably will need to be replaced by the biological products or the microbial products. And this has then significantly driven the investment both by the government agencies, but also the multinational and the startup companies. And there is the real excitement is there. There's a lot to be discovered in the microbiome tools in agriculture. For example, if you look at the human medicine, between 50 to 60% of human medicines have a natural origin, either through the plants or, and majority of them from the microbes. But if you look at the chemical pesticides we use today, is only 11% of pesticides have a natural origin. So there's really a lot of things there that are ready to be discovered and employed in the agriculture. And that has driven the microbiome industry in really a really accelerated growth, from farm productivity to food security and quality of the food to human and environmental health. It is one of the fastest growing industry in the world today. But the use of microbial inoculants in the agriculture is not new. We have been using for decades. In fact, rhizobiomes have been used more than 100 years ago. But the problem is the efficacy of the microbial inoculants in the fields are inconsistent. It works in some ecosystems, some soil, some farm, but does not occur in others. If you look at the product availability, there's a lack of product diversity. If you look at in what is in the market today, it mainly comes in bacteria. It mainly comes from either from Bacillus genus or now some in the Streptomyces. Otherwise, very few from other bacterial taxa. In fungi, it's a Trichoderma and a couple of more, but there is a limited number of the taxa that is being used either as a growth promoters or as to control the pest and pathogen. The third thing is the sustaining the activity of microbes. Microbes aren't chemical that you leave it and after a certain period of time go in through. The formulations have to be such a way that microbe survives. This is a real constraint. And there are other factors that can influence the activities we did not talk about, is the competitions and the desiccations, the fermentation, so on and so forth. But another example is there's a lot of product in the market, but there's no regulatory agency in the regulatory framework to distinguish, particularly what we call the plant growth promoting microbes, to distinguish between genuine and the snake oils. And this then requires to have a better understanding of the plant microbiome assembly. I will talk about that in a minute. Why? Why? One of the reasons is there is a significant knowledge gaps in the fundamental and applied side of the plant microbiome interactions. Fundamental, I think we talk about that one of the things we do not understand, the relations, eco-evolutionary relationship, that are evolutionary process that determines the microbiome assembly, particularly in the plant root regions or inside the plant tissues. We have a very poor understanding of what are the healthy and unhealthy microbiomes. So we say there's a healthy soil, there's unhealthy soils, but we have no understanding what this means in terms of the microbiome and any biological activities. Distinguishing correlations from the cows. And if you look at another aspect that we have a little understanding is the microbiome beyond rhizosphere bacterial communities is very less known, other than some of the fungal side of things, both in the gut and microbiomes, just like in the animals, also in the plant. And another terms is the applied research gaps, and where in fact is that inoculant industry have been using this organisms, isolated from the microbes, fermented it, put in formulation, throw back in the soil, and expected them to govern. But these microbes is going into a foreign environment, alien environment, and they need to compete with the indigenous microflora and have to able to utilize the resources that is being available. But to have a success, we need to understand the ecological requirement of those inoculants for the colonizations of the new environment. Unless we understand what are those ecological requirements, it will work in some area where the condition is favorable and it will fail in other area where condition is not favorable. We also need to understand the interplay between the introduced microbiota with the host genetics and immune systems. We have a very good understanding in animal model, but also increasingly in the plant model, that the plant physiology and plant immune systems play a very important role, which microbes will be able to colonize its surface or inside the tissues. And we need to understand better for the microbial inoculants to grow the plant. First thing it needs to do is to colonize the plants. And we need to understand how that introduced microbes then interacts with the plant immune system. And there's other sort of things that can help going forward. One is the tools to manipulate the indigenous microflora. If we have an ability to switch on activities of some of the plant microbiome without introducing new microbes or just by providing some chemical signals, that can achieve a lot better results than other way around. So to address these issues, what we proposes is what we call the system-based approaches that simultaneously look into two aspects. One is the eco-evolutionary interactions between plant and microbes, but not with all microbes, what we call the core microbiome. And the core or the keystone microbiota. And we defining the core microbiomes are those that are present in a particular plant species in all or most samples, irrespective of the climate conditions, soil types, and management practice put in the perspective. And because these microbes are considered to be inherited, either vertically or have ability to attract those selective microbes wherever you grow them. So they are dominant in this most of the environment. And if you put that plant specifically will attract that microbiome. So there is a mutual recognition between the plant and those microbiota. And so if you target those microbiota as a bioinoculant, then the chances of those microbiota to colonize the plants is significantly higher. As a result, your phenotype will increase. And we also say that there is other aspect that need to look into the multidisciplinary research, public partnerships, and coordinated global approaches. But does the core microbiome exist? There is now significant evidence it does. I'll just give you one example that we looked at in the sugarcane across Australia. So we sample around 1,600 kilometer radius where all the sugarcanes are grown, and we look at the leaf and root and rhizosphere core microbiota. This is just here example that if you look at there's only 0.2% of phylotypes or OTUs are fit into that sugarcane microbiota core microbiomes. And if you look at the relative abundance of those 0.2% is 35%. So there's only few species or phylotypes, that just satisfy the core microbiome definitions, but they are dominant. And that goes both for fungi and bacteria, both in leaf and roots. One of the questions that people always ask, and then again, you're still talking about the hundreds of species, and then we use the network analysis to identify the hub microbiota. And what is the hub microbiota? Microbiota that is a link to most of other, statistically at least, most of the other species or the phylotype of that leaf or the root systems. And we come back with the 12 species or phylotypes of the microbes. There's always questions that what is this statistical analysis? And you can't have cause and so this is the correlations, and you can't say this is have a role in the plant physiology. To test that, what we did, we take out one of the phylotypes by using antibiotics and grow the plants. What do you see here? There's a 50% decline by just knocking out one of the hub microbiota from the sugarcane. That gives you the evidence. This microbiota is not there just because of a statistical dominance, but it have a critical host functions. And if you remove one, it have a direct consequences for health and fitness of the plant. So that's really exciting news. But I guess we need to, and as I said, that another excitement comes from what we have understood in terms of the human microbiomes and how the human microbiome influence every aspect of the human health, including our physiology, metabolism, disease progressions, our behavior, and so on and so forth. Plant microbiome do the similar role, but our understanding of the plant microbiome is very limited in comparison to the human microbiome. We have a little understanding beyond the rhizosphere microbiome, although the research is increasing to look at the endophytic microorganisms. And as I said, that the microbiome beyond bacterial communities, except for the EM fungi or ectomycorrhizal fungi, the mycorrhizal fungi is limited. And we need to move beyond that. We look at the fungi, the whole fungi, but viruses and other things like protists, they also play an important role in the plant health. So moving forward, what we believe that we need to have a system-based approach, including predator and prey relationship, and the public-private partnerships for the research inception to market, and the coordinated global approach. I think I said that before, approaches for the improved outcomes. And to address these particular questions, we have established what we call the global initiatives of the crop microbiome and sustainable agriculture. The short-term aim is to define the core and hub microbiota of these crops, and what are the processes that influences the colonizations of the rhizosphere and the plant endophytes, and what are how these microbes then contribute to the overall performance of the plants and the yield. And the long term, of course, have a policy relevance in terms of the economic growth. It will create a new industry in the inoculations. It have a critical role in sustainability and biodiversity, climate mitigation, and so forth and so forth. So what we are doing in this project, we have identified 5 crops. That these are the wheat, rice, corn, potato, and cotton at the first instance. For each crop, we will sample 250 sites across the globe. And then we'll sample the rhizosphere and the bulk soils, but also the natural sites of 250 sites. And then for each plant samples, we'll have a leaf, a stalk, and roots samples. And then we will do the microbial analysis along with the soil properties, soil functions, plant nutritional properties, and plant traits, and so on. And this activity is already in the 2nd year. Currently, we have 178 scientific groups from across the globe who have signed us to work with us to drive this research. You see the distributions at this moment on the paper. It looks quite good. In fact, we already have received samples from the 27 countries. Our target is 73. And we are working in a partnership with the multiple organizations, including the Global Soil Biodiversity Initiatives and the Microbiome Support and Biodigests and Agriculture Microbiomes. I believe the Linda Linkel is speaking in this atmosphere. So with that, I would like to finish my presentations with the acknowledgement of the key people who have contributed to some of the data I presented today. On the top of the slides is my external collaborators, comes from the USA and Europe particularly, and the bottom 2 lines is the postdoc and PhD student of my lab who carried out most of this study. Thank you very much for your time, and I wish you have a wonderful session of this symposium. Thank you.
Okay.
Dr. Singh. we will not be taking questions, of course, on his presentation. We'll move on with the next presentation by Dr. Zaura. She is a professor of oral microbiology, oral microbiology at Egija University in Amsterdam, and her research is focused in the oral microbiology in health and disease, and how to translate this knowledge actually into clinical practice. Today, her talk is titled "The Oral Microbiome: Oral Health, Resilience, and Systemic Health." Just reminding everyone, please write your questions in the question box. And Egija, the floor is yours. You may begin.
Thank you. First of all, I would like to thank organizers for inviting me. And I see that the time is running, so without due time, I will continue with my talk. First of all, we'll start with disclosure or conflict of interest. So research which we perform at ACTA has been sponsored by several companies, as you see them mentioned here. An outline of the talk today will be about focusing on what do we mean when we talk about healthy oral microbiome. We'll also show that it's quite heterogeneous, even when it's in healthy young individuals. And what happens when we expose oral microbiome to environmental stress. And I will also touch upon oral systemic health link. So what is a healthy oral microbiome? There is no one single definition, just like in previous talks about the soil. It depends on which stage of your life you are in. If you're just an infant, at the beginning you don't have any teeth, and then your first teeth erupt, and those are deciduous teeth, and then you get mixed dentition, and then you get permanent teeth, and then you get maybe braces. And then if you get older and you are unlucky, you might even lose your teeth and you have dentures. So each phase of life will bring also a different ecology in your oral cavity. But in general, we always will have bacteria, and we will have fungi. We will also have viruses. Majority of those are bacterial viruses or bacteriophages. We also have protozoa. So depending on how often a person cleans teeth and depending on water source, you can have quite a lot of protozoa because they feed on bacteria in your mouth. And besides those, we also can have archaea, which are usually the only methane gas producers. So they're not only in the gut, but they're also in the oral cavity. Every day, we actually harm a lot our oral microbes. We do everything to remove them, to kill them, to perturb them by cold and heat foods and foods containing antimicrobial substances, toothpaste and mouthwashes containing antimicrobial substances, but also our own saliva having immune components and antimicrobial components. All these attack continuously our microbes. So you might think that every day we wake up with completely new oral ecosystem, that it changes due to all these perturbations. Well, Human Microbiome Project, which had been mentioned already before, collected microbes from different niches in the human body. And in part of the individuals, they collected these microbes at 2 occasions with few months in between. And then they looked at similarity or the correlation between 2 related samples. What you see here is a Spearman correlation coefficient. So when it's high, the samples are very similar. And here are the different niches they took and the number of individuals these niches were collected on. And as you see here, the least reproducible niches are places on all over our skin, but also vaginal tract, also the nose. And when you see here in red, these are all samples collected in oral cavity. And the green here in between is the gut. And the champion, actually, the most stable microbiome in human body appeared to be tongue. So no, we're not waking up every day with a different oral microbiome. There's this very specific stability of the oral microbiome. We also tested what happens to human microbes in the gut and in the mouth when we expose healthy individuals to different antibiotics. And we followed these individuals for a year. And we looked at both feces and saliva right after antibiotic exposure, after 1 month, 2 months, 4 months, and 1 year. And here you see part of the data where we compare placebo, ciprofloxacin, or clindamycin groups. So it was randomized clinical trial. And here, similarly to the previous graph, the higher the value, the more similar the samples are. And here we compare the baseline samples with right after antibiotics. Baseline was 1 month, 2 months, 4 months, and 12 months since antibiotic exposure. The green triangles are the placebo samples, fecal samples in this case, and you see that the similarity drops from 0.8 to 0.6 in time, even if you don't expose to any antibiotics. And we were really shocked by seeing the results, what happens when you expose healthy individual to clindamycin, because the similarity of the baseline and the consecutive samples actually dropped and stayed low during the whole year. And it was not significantly different from the placebo after 1 year, but not all individuals had reached the complexity of microbiome they had before this exposure. And imagine if these were sick individuals, or these were individuals receiving antibiotics more than once. So, but today we were not supposed to talk about gut, and the reason I'm showing this graph is actually that we also looked at saliva in the same individuals at the same time point. And what you see here was also striking, that almost nothing happened. Also, the green triangles, which are placebo saliva samples, they were stable, they didn't drift like the gut. And when you see the clindamycin, which was so dramatic in the gut, actually only had some effect right after the antibiotic exposure, and after 1 month it differed slightly from the placebo, but further on nothing happened. So again, this confirms the high resilience and stability of oral microbiome. But then I said I would like to actually address the question, what is then a healthy oral ecosystem? We have these healthy microbiomes, but what is the whole ecosystem? What makes it healthy? And usually, I mean, my background is dentistry, and as a dentist, if I look into somebody's mouth and if these squares are individuals, I can say that these are really clinically healthy. I can say that these two have caries or tooth decay, and these two have periodontal disease. But then sometimes I'm in doubt, and I cannot really tell if these people are at risk of some disease, and that's what I would like to say. So we set up a study with the aim to describe actually the boundaries and the heterogeneity of healthy oral ecosystem. And we wanted to assess the relations between the bacterial composition in saliva, the metabolites in saliva, and the related biochemical salivary parameters and the diet of individuals. And we looked at almost 300 healthy young individuals who actually were collected. We collected all types of samples, and part of this data was published in ISMA journal. So I will just show you the summary of the study. If you want, you could read the study yourselves because it's open access. So what appeared, so here in a corner, you see the heat map of the microbiome. And as you see here, these different squares, we found several groups or ecological types of microbes. And what also appeared that they correlated with certain metabolites and certain biochemical parameters of saliva. And what was most striking, that there was certain dichotomy in our groups. So some ecosystems were more saccharolytic, or breaking down sugars, and some were more proteolytic, breaking down proteins. And it was related to different bacteria and different metabolites. And then also we found that some individuals were more specialized and some were more adaptive. So we ended up with these 5 different types. And these specialized, here on the left side, you see they were really specialized in breaking down sugars. And these on the right side were specialized in breaking down proteins. And these in the upper parts were actually doing everything right. So they had microbes which were adaptive. They could do both. And what we actually think, and this was a cross-sectional study, so we cannot confirm at this stage, but we think that these 2 groups here at the bottom might be at the early stage of dysbiosis, so really losing the balance because they're too specialized. But remember, when we looked into the mouths of these individuals, they were still healthy. So we call these ecological types or oral ecotypes, and what we actually think that if this is true, that even if we are clinically healthy and we are all different, one size does not fit all. So we all will need a different oral care according to the ecological type we carry. And we also think that these most specialized ecological types could be more susceptible towards disease or could indicate a disturbed ecosystem at the preclinical level. So one size of prevention does not fit all also. So if we are healthy, but we would all use the same toothpaste or the same preventive tools, it might not be the best for oral ecosystem. So the challenge for us, for oral research professionals, is to identify which are the right individuals for the right preventive strategy. Let's look what happens when ecosystem is exposed to these daily perturbations. So like I said, we are having this complex ecosystem. Imagine this sphere being oral cavity, and it goes through the trajectory of somebody's life. And then it's exposed to different, there are different factors, and these factors are exposed to different stresses. So I illustrated the factors as springs, and it shows the resilience of ecosystem. You can press the sphere and it will resist the stress and it will inflate back. So basically, this is what happens every day when we chew, when we expose our oral microbes to different treatments. So they can handle this because all these issues are collaborating in this stability. But we also wanted to see if we are all equally resilient and what happens if we expose ecosystem to quite severe stress. It was all ethically approved. We performed an experimental gingivitis study, which is a very accepted protocol for clinical studies. So you ask volunteers actually to abstain from toothbrushing or from any other oral hygiene measures. So these individuals, we assess them clinically at the baseline. We actually duplicated the baseline so that we have replicate measurements and assessed the 2 weeks after abstaining from toothbrushing. And then also we gave them time to recover and we assessed them again. And in this time, we also took samples for microbiome analysis, and we also took images, which we call QLF, quantitative light fluorescence. So I'll explain you in a minute. So this visualization of dental plaque is quite specific measurement for dentistry. You can color dental plaque like you see on the right picture. So it's colored with staining solution, which stains very young plaque pink and all the plaque dark blue. So you see this dark blue line here, and this is corresponding white light image without staining, and you can see that there is a dental plaque here because this person did not brush for 2 weeks, but it's hard to see. What you also can do, you can take a picture under specific light wave. We call it fluorescent image. And you can see here without any staining, I hope you are in your screen resolution allows seeing it, that you can see here this red layer, which corresponds with this blue layer. So basically what we see here, that old mature dental plaque fluoresces red under specific light. We used this property in this study, so we also took these fluorescent light images throughout the experimental gingivitis study. So what you see here are selected individuals, A, B, and C, at the first day before they abstained from brushing, so they were supposedly clean. And you see here, I hope here at the bottom already, some red fluorescence appearing, and also here you see red fluorescence in individual B. So when these individuals continued with the study, you could see that individual C actually started fluorescing quite a lot. And if you continue, it even became like glow in the dark individual if you would see this person somewhere under fluorescing light. While hardly anything actually happened in the individual A, even though this individual also didn't brush teeth. And we checked it because you could see in the white light that there is dental plaque, but this dental plaque didn't become mature and didn't become fluorescing red. So when we analyzed, we could quantify these fluorescence images. So you could see that there were, we could split our individuals in 3 response groups. So this group with very low fluorescence change, this intermediate, and this very high. And actually what was also interesting, the response was high in this group, but they were also having very high red fluorescence values at the beginning, at the baseline. So they differed already even before we started this intervention. So what we actually also found that when you continue such a study, the whole idea of experimental gingivitis is that after 2 weeks, you create an ecosystem which is diseased. These individuals should have bleeding gums, but not everybody became inflamed. So not every ecosystem collapsed. And here, when we correlated the fluorescent plaque after 2 days of not brushing, so at the beginning of the study, it already correlated with bleeding at the end after 2 weeks. So basically what we see here, that by measuring this fluorescence, we could already predict who would really be harmed by not brushing. And when I put here this picture of dentist appointment, I mean there that before going to dentist, most people actually do their best. They brush, they floss, they rinse, but they should not do so, because by coming without brushing, it gives us opportunity to predict the risk for inflammation, at least in this case. So with this example, I wanted to show that we exposed these individuals to the same perturbation, but they differed extremely in their responses. And I only showed one part, this red fluorescing plaque, but they also differed microbiologically, they differed in their bleeding, so they differed in many aspects. And you could see that, yeah, the ecosystem is not equally resilient at each ecological type. So maybe this also is related to the host ecotypes we discovered in the previous study. But that's again something we are investigating currently. Well, in the last part of my talk, I will discuss the benefits to the host and interaction with the whole host of the oral microbes. This is an example from Sweden, where they had a study on small infants, and they asked parents, what do they do when the infant uses pacifier and the pacifier falls down on the ground? There was part of the parents who always meticulously cleaned the pacifier and cooked it, and there were parents who were licking it like Britney Spears here. And they followed these families in time. And here you see microbial communities. It is PCA plot. Each triangle is one saliva sample. And here you see cluster of salivas from pacifier boiling families from the infants, and here from pacifier sucking. So this was not a surprise that by inoculating your child with your own microbes, your child creates a different microbiome. But what was more interesting, that when the children reached 2.5 years of age, the children who were exposed to salivary microbes of their parents by sucking the pacifier had lower risk for eczema, asthma, and sensitization. So basically, their immune system was trained and more mature and developed healthier than infants who were not inoculated at a timely manner. Another example is, for instance, the oral microbes. When they're there, they protect us from intruders. So they keep the niche occupied so no other microbes can sit there. But they also, when they are killed, the niche is available. An example of that is outgrowth of opportunistic pathogen like Candida albicans. It is a very painful and debilitating condition, this white part on your tongue. When the microbes or bacteria are killed by antibiotics, then these opportunistic pathogens, fungi, can proceed and grow out of proportions. Well, the third example on benefits of oral microbes will be about nitric oxide. As you might know, this is a very active, physiologically active molecule, and I will focus on its role in vasodilatation. So if endothelial cells release nitric oxide, the blood vessels dilate and your blood pressure goes down. But how do we get nitric oxide in our blood? Well, mainly it is from nitrate-rich foods. These are leafy vegetables, and these foods, so they contain nitrate. It has to be reduced in nitrite, and then in your stomach it is reduced to nitric oxide. There's one problem, that's this red arrow, because our own cells don't produce enzymes for this reduction. So what we need oral bacteria for this step. And these oral bacteria do it, but they cannot do it at the moment while the food is still in mouth, because that should be a very quick job then. So basically they get chance to do it slower, because nitrate goes through stomach, intestines, and is absorbed into blood and is reabsorbed and concentrated into salivary glands. And while we are doing something else 2 or 3 hours after the meal, this nitrate is released in the oral cavity, and oral bacteria can reduce it to nitrite. And does it work? Yes, there are several studies. I'm showing you only the oldest studies, the pioneering studies, which tested the nitrate supplements and actually showed that supplementing a diet with nitrate protects your cardiovascular system and reduces your blood pressure. You can also inhibit this effect by killing oral microbes using antiseptic mouthwash. So again, it was indirect proof that oral bacteria are involved in this process. When we look at oral ecosystem, I said it is different at each stage, but when it's healthy, it's definitely in balance with the host. So they perform these beneficial functions. I just showed some examples. However, things go wrong if things are losing the balance. And for instance, here, if somebody snacks too often sugar-containing snacks, then you can result in very low diversity aciduric microbiota, and you can lose your teeth. Or on the other hand, if there is an overruling inflammation and these pro-inflammatory bacteria grow along the gums here, and the person is susceptible to these pro-inflammatory molecules, then you can lose the bone, which keeps your teeth in mouth, and your teeth can become loose. If that would be all the problem, it wouldn't be a big issue because you could also extract the teeth and have a denture. But the problem is that all these inflammatory products actually end up in our bloodstream. They end up in bloodstream and end up in different parts of our body where they don't belong. And not only these pro-inflammatory products, but also bacteria themselves can enter the bloodstream via the gums here. They can easily into the bloodstream. And here I will show you some examples of the so-called mobile oral microbiome. So oral microbes have been associated with very many conditions, debilitating conditions such as cancers, but also with brain tissue and Alzheimer's disease, but also with pregnancy complications and rheuma. So these are just few examples, and I don't have time to go into details. So there are several reviews on each of these where they explain how this could happen, that these microbes actually have such an impact. The causality is not proven, but there is certainly association. So if you have colon cancer examples, for instance, they do find oral bacteria such as fusobacteria in this cancer tissue, but they don't know so far if these bacteria initiated it or they actually went there when the situation was already out of hand. Of course, we don't want to have these disease-producing microbes. We do want to modulate them and keep our ecosystem healthy. So there are different ways how to modulate it, like using pre- and probiotics, or exchanging your microbes maybe, or transplanting them from the healthy individual. But can we actually modulate it? So my final slide is actually about the challenges. Of course, many of you have heard about fecal transplants, which are quite successful in certain cases, like Clostridium difficile infections. This, however, would not work that successfully in the oral cavity. And just few reasons why these are big challenges to modulate. Because as I showed you at the beginning, this extremely high stability of the oral ecosystem, it will actually work against any changes we would like to introduce. And so you would first have to remove all existing microbes. So that's also this colonization resistance. If the site is already occupied, there is no place for newcomer. So how to remove all existing microbes and how to prevent that opportunistic ones will start growing out. And lastly, like I showed you, even if we're all healthy, we are all different, and our own intrinsic properties like salivary pH, different enzyme activities, will affect which microbes prefer our oral ecosystem and which won't. So it's not like you can have a probiotic strain and implant into somebody's oral cavity and it will grow there. So indeed, there are a lot of challenges. And take-home messages for you from this talk. Healthy oral microbiome is very stable and it is resilient to stress. Well, maybe not in everyone. Like I showed you, some people suffered more from stress than others. Healthy oral ecosystem is highly heterogeneous, so one type of prevention will not fit us all. And healthy mouth is a key to a healthy body, so please take care of your oral microbes. And with this, I would like to thank the collaborators for the large study we performed on healthy individuals. and would like to thank my group at Department of Preventive Dentistry, and thank you all for your attention.
Thanks again, Egija. Great presentation. I would like to open the questions with a question that actually occurred to me. You presented a very nice data that some people never go on to mature plaque. Have you looked at all into quorum sensing, communication happening there? I mean, often quorum sensing is responsible for maintenance and forming biofilms. So I was just wondering if there is inherent signals that are not present on those people. Your microphone seems to be off.
Muted myself because I thought I should. So yes,
No,
the samples, of course, were too small volume to measure quorum sensing molecules. But from the studies on biofilm interactions, we do know that you need quorum sensing also to maintain stable community. So your hypothesis could be true that these people might be missing something, but it also could be that they have very active quorum sensing, like warfare even, and because these inhabitants at normal condition keep this balance, they actually won't let the pathogens to grow out. So they really have fight with outgrowths of these anaerobic bacteria, which we usually see fluorescing red. We don't know. So both could be true, nice and interesting to test, but I think we should do it not on humans, but in vitro, vitro models.
Sure. Thanks. There's a question coming here from the audience. So are there any studies of oral microbiome after modulation with probiotic strains? Was there any shift in microbial ecotypes? Actually, I was curious about that too, whether that kind of intervention actually does anything.
Yeah, I wish I had 1 more hour, then I would have shown you work we've done on probiotics. There are several studies on probiotics. The major problem is, I said, like this stability. Another problem is the choice of strains to use as a probiotic. So far, strains in the market meant for oral care are actually originating from foods and are typical gut strains, like mainly lactobacilli. And its oral cavity is not their natural habitat. So they will not nest there. They won't become naturals. It's a very unusual situation for them. So I think the future is actually in probiotics originating from oral cavity. And there is some research ongoing when really super healthy individuals are screened and strains have been identified which have interesting capacities like arginine deaminase activity, so that would increase pH for the cases when you have caries problem. So inducing these bacteria introducing them into somebody's oral cavity would keep neutral pH instead of lowering it down. So it would reduce caries risk. And the same would go for pro-inflammatory bacteria, that there are several bacterial toxa which can inhibit these pro-inflammatory ones, but they should be selected from the pool of bacteria which you can identify not naturally in oral cavity. That means they have properties to attach to teeth, to mucosa, and outcompete others, because otherwise the chances are very limited, unfortunately, for the manufacturers.
Sure. We have time for one more question. I'll select one here from the audience. Well, one is rather quick, perhaps. It's just a question on oral methanogenesis. How common is it in people? Does everybody have, or do we know the frequency in the population for oral methanogen?
Great question. It's one of the forgotten inhabitants. So there are no large population studies investigating it. There are some focused studies on people with periodontal disease in comparison with healthy. And they are found both in people with disease and people with health, but in higher abundance in those with disease. But they are not really pathogens. they are associated with pathogens. So the hypothesis is that they promote growth of periodontal pathogens by reducing the environment. So but they are in everybody's mouth, so it's not like you can be immune to methanogens. They like anaerobic crypts of the tongue, and these can be very deep, so they can have very cozy environment there to grow. You don't have to have periodontal disease to have archaea.
Great, thanks. Okay, we are getting advanced on time, so I think we'll probably move on. Thanks again, Egija, for the presentation. It was very nice. With that, we'll move on to our next presenter, Dr. Philip Bernat. Is an associate professor at the Department of Psychiatry at the University of Oxford, and his research aims to explore novel molecular and nutritional interventions that can assist in the treatment of psychiatric disorders. Today, he will talk to us about this interaction between gut-brain axis. His talk is titled Prebiotic and Probiotic Modulation of Cognition and Emotion. Dr. Burnet, please.
Thank you very much, Chris. Can you see my slides?
Yes.
Yeah, oh, okay. Is it blocked by the other things on the right? I seem to be having trouble with my laptop. Okay.
Are you? Yes. Are you good now?
Yeah. Can you see? Can you just see my slides, or can you see the other boxes on the right?
No, we can see just the slides. You're good.
Excellent, excellent, good, because I'm seeing something completely different. Okay, well, thank you so much, Chris, for your kind introduction, and thank you for the to the committee for inviting me at this excellent conference. I know time is moving on, so for the audience sake, I know I'll try and make this quick, because I know some of you will be approaching lunchtime, some of you will be approaching dinner time, and some of you will be wanting to go to bed. So I think I'll just start by saying what I'd like to do is talk about how we used prebiotic and probiotics supplements as tools to test whether gut bacteria could actually modulate brain function. Now, this is not only a way of determining whether there are other peripheral mechanisms that modulate our brain, but also to develop a potentially new treatment for psychiatric disorders, especially those which are resistant to current therapies. I should say I am a neuroscientist, but I work closely with psychiatrists, but still am quite sane. So just my conflict and interest, some of the work is industrial funded, a lot of it is from academic funding. So why look at pre and probiotics? So first of all, I'll just mention very briefly why we were interested in looking at gut bacteria and brain modulation. Then I'll go into the prebiotic effects specifically of cognition, the higher function of the brain in rats and humans. And then I'll go on to probiotic effects, which is more of the more recent studies we've done, particularly on emotional behavior, and I'll discuss that. And then go on to how we looked at probiotics and low mood in humans. So why are we doing this? Why are we manipulating gut bacteria to look at the effect on the brain? Well, the influence of gut bacteria on host metabolism and immunity has been known for a long time, but it wasn't until 2004 when Sudo and his colleagues showed that germ-free mice had a high response to stressors. Okay, so they were highly stressed, anxious animals in the absence of gut bacteria, as opposed to their counterparts, control counterparts, which, when exposed to a stressor, showed a normal response. So these were hyper responsive to stress. Now, we were interested in this in particular because not only did they show the stress response we exaggerated in these mice, but also in the brain, the glutamate Nmda receptor subunits and obligatory subunits, the NR1, NR2A, were reduced in the brain. Now, I'm particularly interested in the Nmda receptor because it mediates. It's important for the maturation of the brain, particularly cognitive function. And its hypofunction has been implicated in pathogenesis of schizophrenia. So my search mainly focuses around ways of augmenting this receptor. And so we started looking at manipulating gut bacteria to achieve this, based on Pseudo's work showing that germ-free animals, a complete absence of gut bacteria, reduced these receptors in the brain. So first of all, prebiotics. Why did we want to go to prebiotics? Well, at the time when we started this, there was already, as you saw after Pseudo's work, literature on how probiotics might affect brain function. But we wanted to go for prebiotics because we wanted to augment the gut bacteria that were already in the gut, the indigenous populations, which there are more of. You can manipulate more gut bacteria with prebiotics than probiotics. Obviously, probiotics are a few strains or multi-strain, but with prebiotics, you hit quite a large percentage of the beneficial bacteria. And a lot of these things are found in things we probably don't like to eat, but nevertheless they're there. And the main one is which is supplied as a supplement, the fructooligosaccharides or FOS, and these are simple short chains of fructose flanked by glucose, as I'm sure most of you know. Now, there are also galacto-oligosaccharides where instead of a fructose, there's galactose. Now, these don't normally appear naturally in nature. These are more synthetic, especially long chains. Of course, you do get glucose, galactose chains in these products, but they're not as long as the synthetic ones which are manufactured. Now, this is interesting because we and others have shown that bifidobacteria are particularly fond of galacto-oligosaccharides. So it is quite bifidogenic, the galacto-oligosaccharides. And we've shown that just feeding rats with galacto-oligosaccharides, look at their fetal bifidobacteria counts, that there are more bifidobacteria in the guts than there are when you feed them with FOS. So this wasn't new. This is something that others have shown as well. But what is interesting with prebiotics is you can get actually two hits. Not only do you increase the population of beneficial bacteria, which may then impact the brain, either by modulating the immune system and or the vagus nerve, but also the prebiotics themselves are broken down to potentially neuroactive substances, the short chain fatty acid metabolites, acetic acid, butyric acid, and propionic acid. So here we thought prebiotics were good because we amplify a larger population of good bacteria already residing in the gut, and also there will be a supply of short chain fatty acids, which benefits the gut themselves. And I'll show you, I'll discuss how they might go into circulation and affect the brain. So we chose the galacto-oligosaccharide because for the reasons that it's more bifidogenic. And our first question was, do these prebiotics increase Nmda receptors? We've seen that in germ-free rats, they decrease, germ-free mice, they decrease. So does increase in gut bacteria increase these? And we did actually find that feeding rats with actually both fructooligosaccharides and galacto-oligosaccharide increase Nmda receptors in specific brain regions important for attention, such as the frontal cortex, and the hippocampus for memory and spatial memory. So these are Western blots showing the changes in the subunit. And we also found that giving the prebiotics increased one of the co-agonists of this receptor, and that's D-serine. So not only did it increase the receptor, it increased the amount of the co-agonist that's needed for the receptor to function, as well as glutamate. So that was interesting. But of course, you know, in animals, these might be a homeostatic response. This doesn't necessarily indicate that these receptors were functional. So our next step was to investigate whether the increase in NMDA receptors in the brain were functional and caused ultimately a behavioural effect on the animals. And in particular, as I mentioned, the NMDA receptors are important for optimal cognitive function as well as neurodevelopment. So we used a cognitive test to see if particularly the BGOS prebiotic improved cognition in rats. Now, but first to do that, we went further upstream and looked at the function of the NMDA receptors. So we did this by using in vivo interferesis, so basically a cannula in the frontal cortex of the rat injects an NMDA NMDA receptor agonist, NMDA itself, and we measured the electrophysiological response of cells in the brain to micro-iontopherese NMDA, so micro-injected NMDA. And we found this is a dose response curve. So you see at different concentrations of NMDA, there's an increase in response following prebiotic intake. So this was a 3-week prebiotic intake, daily ingestion of this galacto-oligosaccharide. So the increase in NMDA receptors actually correlated with an increase in their function. But we also went further downstream, as I said, to see if this translated to an improvement in behavior. And lo and behold, we did actually find that the attention or cognitive flexibility of rats was increased. I won't go into details explaining this test, but basically cognitive flexibility, as most of you will understand, is adapting to new changes in the environment. Okay, so you're flexible about adapting to your surroundings. So this is the important part. Animals who are under control, they take so many trials to find a reward in a task. But those with the prebiotic didn't take very long to work out where the reward was. Obviously, there's obstacles and so on. They have to work out where the reward is by figuring out what's changed and what hasn't changed in their environment. So prebiotic feeding, particularly the galacto-oligosaccharide, which increases bifidobacteria, increase NMDA receptors, which increase in function and increase cognitive flexibility. And this specific function of the brain is centered around the medial prefrontal cortex. So that's an important executive function that we all need, that makes us human, if you like. So very briefly, what could the mechanisms be? Well, as I mentioned, that prebiotics are broken down by bifidobacteria into short-chain fatty acids, acetate, butyrate, and propionate. Now, propionate and most of the butyrate is taken up by the gut itself and uses an energy source. But a lot of the acetate which is produced does actually go into the circulation. And several studies before us showed that acetate uptake, there's an interaction between NMDA receptors and acetate uptake in the brain. And indeed, pharmacological levels of acetate injected into animals reverses blockade of NMDA receptors. So if you give more acetate, NMDA receptors seem to function more. So this might be a mechanism by which the prebiotics had their pro-cognitive effect in our animals. The galacto-oligosaccharides broke down, produced a lot of acetate that flooded the brain, increased NMDA receptors, and then led to the increase in their transcription function and so on. And the mechanism, as I mentioned with transcription, acetate, short-chain fatty acids are been shown to be histone deacetylase inhibitors. So when you inhibit histone deacetylase, yes, inhibitors. When you inhibit HDAC, you increase transcription. So this is the hypothesis where we followed that the increase in acetate might increase transcription of the NMDA receptors. However, as I emphasized, these studies have been done at pharmacological levels of acetate. What we did, when we gave the probiotic to animals, we did see an increase in plasma acetate after prebiotic feeding, and we also showed an inhibition of HDAC activity, as has been seen with pharmacological levels of acetate. However, when we injected acetate to make plasma levels comparable to prebiotic levels, not pharmacological levels, so it's 10 times lower than pharmacological levels, we didn't see an inhibition of HDAC activity. So the prebiotic reduction of HDAC, which we certainly saw, was actually acetate independent, even though it does the same thing as acetate. And we hypothesize it must be the acetate. It did seem to be, we couldn't replicate it at the levels produced by the prebiotic. So we probably, we're looking into other mechanisms, as I said, looking at the vagal gut connections and the role of the microbiome itself. We didn't actually measure microbial levels as such. So as I said, a lot of this work is mainly because our interest in improving treatment of schizophrenia, which is, affects 1% of population, is quite a severe mental illness, and there is very little treatment for that. There's psychosis, which can be treated in 70% of population of psychosis patients. 30% are treatment resistant. There's negative symptoms such as withdrawal and isolation and so on. But the thing that always occurs with schizophrenia or early psychosis is a cognitive impairment. And this is thought to be arising from a hyperfunction of NMDA receptor. So this fits in with what we're doing. We found a way of increasing NMDA receptor function, so could this work in schizophrenia? And of course, more recently, there has been links with the microbiota associated with psychosis, symptom severity. So I should say that the reason why we are interested in cognitive impairment is because this is the factor that really stops people with psychosis functioning. So even when they have medication and they're not psychotic, they still find it difficult to function in society, sustain a job, keep relationships because of their cognitive impairment, or even decreased cognitive flexibility, as I mentioned. So what we did was we wanted to see if our findings in the animals, in the rats, translated to human population. So as you can imagine, we recruited people with schizophrenia, first episode psychosis, and we measured their overall level of cognition, and we gave them a prebiotic, the Bigos prebiotic, to take for 12 weeks, daily for 12 weeks, and then after that they would take a placebo. So this was an open label study. So some started off with the Bigos and then would change to the placebo. And this is, if you look on the right here, B, you'll see those who started on the Bigos, there was increase in learning, in cognition. And then when they were swapped to placebo, there seemed to be a decrease, whereas the placebo group, when they were put on the prebiotics, seemed to have sustained their learning. Obviously, there's an overall learning effect, but certainly even in the presence of a learning effect of the cognitive tasks, you could see that the prebiotic improved cognition. And when you collapse these data, look at group comparison, you will see that the prebiotic overall improved cognition in schizophrenia. So that's quite exciting, and we are trying to replicate this with a larger number and more measures of the actual microbiome. The thing I should say is that it didn't affect any metabolic immune parameters. As I said, even acetate, it didn't seem to change. So we're not sure what's going on there. We're going to look at the microbiome itself and see if there's any clues there. And as the gut bacteria and host immunity are inextricably linked, we obviously measured some plasma immune markers. But again, we didn't see any telltale signs. I should say these, of course, were medicated. So whether that was a compounder, I don't know. But in the presence of medication, there seems to be an effect of prebiotic. Perhaps the prebiotic augmented the effect of the medication. We don't know. We're looking into that at the moment. So what about, so that's prebiotics. What about probiotics, the live cultures which were mentioned in the previous talk? Well, since the pseudo study, there have been many articles looking at rodent prebiotics and anxiety. Anxiety is quite easy to measure in rodents. A bit more difficult to measure depression, really. Which we'll discuss, and obviously impossible to measure psychosis. We don't know if rats hear voices or squeaks or whatever. So that's the so that a lot of work has been done on this. So we didn't really pick this up here until after our prebiotic studies. And because we wanted to make it translational, we were following a systematic review, which a recent systematic review concluded that the more convincing data on pre and probiotics are actually with multi-species probiotics for depression. So not so much mood in healthy volunteers, which a lot of studies have done, including ours, but actually when there's a pre-existing deficit. So clearly with a pre-existing deficit or a model, we're more likely to see the effect of the probiotic. So what we did was we wanted to check the anxiolytic and antidepressant effect in both rats and humans. Now, a lot of the literature, there's quite a few labs doing animal studies, quite a few labs doing, or fewer labs doing human studies. But we wanted to see if the same probiotic in the same lab had the same effect in rats and humans, or similar effect. As much as can be translated. So what we did, we measure anxiety using the light dark box, where anxious animals spend longer time in the dark area of this test box, and we measured so-called depression with the 4-swim test, where animals are put into some water, they naturally swim. And the faster they stop swimming, the greater the indices of so-called depression, or in this instance, maybe learned helplessness. So it's a bit controversial, but it is a quick test to see, and has been reported to be successful, showing antidepressant effect. But anyway, we gave a multi-species prebiotic, a fifth of the dose that's usually reported for mouse studies, in a strain of mice that has been shown to be anxious, an anxious type of strain. We found that after 3 weeks of study, the animals seemed to be less anxious, spent more time in the light, and their latency to go out into the light was reduced. So they were less anxious after 3 weeks. After 2 weeks, didn't see much effect, so maybe it needs time to have an effect. But in the 4-swim test, we didn't really see an antidepressant effect. There was no change in latency to float or time spent floating. The greater the number of time floating, the more depressed, if you like, the animal is, or conversely, the less time floating, the less depressed the animal is. So we found in the animals this multi-species probiotic, which contained bifidobacteria, lactobacilli combination. I won't say the commercial product, but this seemed to have an anxiolytic but not antidepressant. Arguably, we could argue that that's not a good test for antidepressant, but like I said, it's a test that has been used for antidepressant drugs. So we were just following what's been used before. Now, we also found on the same, consistent with our interest in NMDA receptors, we did look at the expression of NMDA receptors. We actually found a decrease in NMDA receptors, which might be actually consistent with studies in humans showing that blocking NMDA receptors has a rapid antidepressant effect. But I'm not going to show those data. I think what's more interesting is that we looked into whether the viability of the probiotic was important. So what we did was we heat killed the bacteria and compared it with, as previously, with the live bacteria. And this is the light dark box measuring anxiety again. And as you can see, animals were receiving the probiotic daily, again, this was for 3 weeks as previously, were less anxious than control, but also the heat-killed bacteria. Those receiving heat-killed bacteria were less anxious. So that was interesting because it showed that viability wasn't important for the probiotic. And it also indicates that perhaps it's the bacterial cell wall that interacts directly with the host gut, which initiate responses perhaps of the vagus nerve and perhaps of the immune system, which then feeds back to the brain. Certainly, depression has been linked to an increase of a mild increase in an inflammatory response, obviously a mild one. So we did look at the immune system, and we did indeed find that interleukin-6 pro-inflammatory cytokine and interleukin-1b were decreased, perhaps more so with heat killed than with live bacteria. However, because this was also seen at 2 weeks, we don't think that the anxiolytic effect of the probiotic was mediated by the immune response. So again, we're looking into more of how we can dissect the actual pathway, the path between the gut and the brain, which is mediating the anxiolytic effect. So what about humans? So lastly, we also looked to see if we could translate our findings of the mice into humans. As I said, in the systematic review meta-analysis, it was shown that the most promising result will probably be from studies of depression and amosphous probiotics. So we certainly we decided to recruit people with low mood according to the Patient Health Questionnaire 9. This is something that primary healthcare uses, and we chose moderate depression because these people were, although they had quite high levels of depression, they weren't receiving medication, they weren't being treated, or reluctant to seek any treatment. So we could actually have a pre-existing deficit without medication confounding any of the data. So we designed a double-blind, two-group placebo-controlled trial, 16 billion CFU per day, or placebo for 4 weeks. We did subjective ratings of depression, so we did a before and after score of depression after 4 weeks, and anxiety with state trait anxiety indices and pre and post. And we did psychological cognitive measures on the last day. Now, the reason why we did this is because when mood changes, the processes that underlie them are actually established psychological processes. So it's not a case of we see euphoria or short-lasting euphoria at the end of the study because everyone was glad the study was over. We actually could measure robust psychological changes in their perception of negative and positive stimuli, which in a way provides information of the psychological mechanisms that underlie mood. And we also measured blood immune markers, as we did with the animals, and unfortunately we couldn't obtain brains, but we did the blood immune markers and the waking cortisol response. So this is the stress hormone that increases when we wake up, and we measured that before and after too. So what we found is that the multi-species probiotic actually decreased significantly depression scores in people with low mood. This indicates significant group by time interaction. There was a slight, as expected, a placebo effect, but this wasn't significant. This was more significant than the actual probiotic. So that fits in with the antidepressant profile. But interestingly, converse to the animal, it wasn't anxiolytic. So these are the readings of these are the results of the state trait anxiety inventory. And as you can see, pre and post, there was no changes between before and after supplementation, or between placebo and prebiotic. So it seemed to have an antidepressant effect, not an anxiolytic effect, which is converse to the animals. I've put this graph in on the right here just to show you what was interesting. Is one of the questions in the patient health questionnaire was about attention, about cognition, and particularly concentration. So people was asked, is your concentration very poor? Can you not concentrate on watching TV or reading the newspaper? And it seemed their concentration increased after probiotic intake. So there is some sort of change in cognitive scores there. But again, it did not affect circulating immune markers, and we didn't see a change in salivary cortisol, the measure of stress response, which is the same as we saw with the prebiotics in schizophrenia. And finally, what about the psychological tests? What are the psychological underlying mechanisms? So we used a test factory of emotional tasks, and basically what these do is measure what our biases. We would subjects are shown pictures, words, and so on, and they score. When they score, we can determine what sort of emotion they focus on, if you like. What specific emotional stimuli they focus on. Now, people with depression tend to focus on negative stimuli, whereas happy people tend to, or even antidepressant treated people, focus on positive stimuli. So as you can see in this graph on the left here, there wasn't much difference between probiotic and placebo, but what did happen was that probiotic people actually focus more on neutral faces. So there was no emotion expressed by faces they see on a computer-based task. Now, as I said, often people with depression, you'll see a reduction in happy and an increase in the negative emotions such as anger, fear, sad especially. But here, and then when they're treated with antidepressants, you see an increase in happy. But here, we actually saw it was neutral. So it seemed to be, they seemed to be unbiased towards their choice of whether they picked a negative, positive emotion. And the same happened in this dot probe task, which is a measure of emotional attention. A lot of this is the placebo, but a lot of people patients who have been on antidepressants actually show this profile. They show an increased bias towards happy stimuli, but the probiotics completely annulled both emotions. So they seem to be like they don't care about which emotion they show. They seem to rather be neutral, seem to be numbed by any emotional stimuli. And interestingly, in a reward learning task, the probiotic, again, they seem to be care less about receiving a reward. Now, this reward learning is informative for for measuring anhedonia, so lack of pleasure in depression. And you can see that so people with depression will have lower win trial and probably higher loss trial, because they get less pleasure out of winning. But here, they didn't seem to care whether they won or lost. So overall, it seemed to be opposite to traditional antidepressants by showing, as we call it, reduces reward salience, or becomes impartial to specific emotional stimuli. So emotionally blunted, they seem to be, for want of a better term. So that's it. So to summarise, preclinical studies showing benefits of gut bacteria appear to be translational. So even the higher functions that we found with the prebiotics, the higher executive functions such as cognitive flexibility, seem to be translational and in severe mental illness. We need to understand mechanisms may not be the same as pharmacotherapies, as we saw in the last slide, or between species. We saw in the animals there was an anxiolytic effect, not antidepressant effect, vice versa in humans. And so we have shown that the pre and probiotics may assist in the treatment of cognitive and mood disorders, but I don't think they're likely to help mental illness on their own. I know we showed that with low mood it may be helpful, which is interesting, because people with low mood not being treated makes up about 50% of the total population of people with depression per se. So that will be a useful treatment to roll out. But I think that perhaps with the gut bacteria improving metabolism of the host is probably more likely to increase the effect of drugs. So and the maintenance of normal health gut may reduce the onset of brain disorders. So you know, we although, like all the other speakers have said, we don't know what a healthy microbiome is. Certainly nurturing the ones we know are beneficial to the gut may help give us some resilience to psychiatric disorders. So finally, I'd like to thank everyone in the audience for your attention, and to thank all my colleagues in Oxford and around the country who've who've put up with me these past few years through my research. Thank you very much.
Thanks, Philip, for this very, very interesting presentation. We'll start with the questions. I have one here from the audience. So just curious to know whether any link between the degree of polymerization of galacto-oligosaccharides and procognitive ability was seen in this study. If, I guess the degree of the chain.
Yeah,
of polymerization.
Yeah. That's a very interesting question. This particular product had a mixture of oligosaccharides, and these are published actually. These ranged from, I think, links of 2 or 3 to about 7 or 9. So we haven't actually thought of fractionating each oligosaccharide to see what contributes to it. But that's a very good question, something that others have mentioned to us about, you know, what is it that's actually driving this? Is it the mixture, or is it individual components?
Right. I have another question here. What about giving metabiotics like short-chain fatty acids, butyrate, directly in schizophrenia and other disorders instead of the probiotics? Multi-species or single species, which one is better? If you could comment a bit on those lines of evidence.
Yeah, so again, good question. So as I mentioned, the studies, the short-chain fatty acids, especially acetate and butyrate, they do have psychotropic effects, but these have been shown in animals at pharmacological doses. So yes, it is perhaps injecting or oral intake of short-chain fatty acids may benefit. But we wanted to find the most natural way of doing this, because indeed it might not just be the short chain fatty acids, as I showed. Acetate doesn't look like it, or butyrate reaching the brain. We wanted to find a natural way through via the gut microbiome, so that metabolism is improved as well as brain function, as we think that brain function may be improved because metabolism is improved. So rather than just shortcut the body and take something that will travel to the brain directly, I think it would be better to explore how we can further look at, we can do it via the gut bacteria rather than take oral short chain fatty acids or inject them, which is basically, another drug approach.
Right.
I don't know if that answers the question.
Well, I guess it comments enough on them. I'll finish with one question from the audience, since we're running a little long on time here. There's a question about what is the actual impact of potential neurometabolites produced by the microbiota, such as GABA, on neuronal function. Could you comment about that type of evidence?
Yeah, no, that again, that's another good question. So studies have been done on lactobacilli, which are GABA producers, but again, the theory is not that they produce these neurotransmitters which then flood the brain, because the brain actively excludes neurotransmitters such as GABA and serotonin. It's more like that these neurotransmitters affect the gut, somehow improve gut metabolism, and then perhaps via the immune system, maybe metabolism itself, or the activity of the vagus nerve, then it influences brain function. The vagus nerve is quite interesting. I think, a study in 2011 showed that a probiotic increased GABA, did increase GABA in the brain, but that GABA receptors in the brain, but that effect was lost when the vagus nerve was cut. So it's not that GABA was flooding in the brain via the blood, via the circulation. It was more that perhaps it was activating or deactivating the vagus nerve and then influencing GABA and other neurotransmitters that way.
Great. Well, like I said, we're running a little late on time.
Yeah, sorry about that.
But that's okay. No, that's okay. It was a great session with everybody. I really would like to thank everyone for participating and presenting. I had a great time. And I also would like to invite all the audience for the next session 3, coming up tomorrow at 11 a.m. as well. The program is on screen for you guys to see. Should be quite interesting for quite a wide range of people. Thanks again, all the presenters, and
thank you.
Have a good day.
Yeah, thank you all.
Thank you.
Thank you, Chris, for chairing the session, and we're looking forward to your participation tomorrow in our innovative packaging session. Thank you again very much. Have a good day. Bye-bye.
Bye-bye.
And you, bye.